Okay, so hello everyone. Thanks for having me here. It's a real pleasure to be in this network. Actually, I already met many very interesting people and also, yeah, very interesting topics. So very happy to be here. And today I'm going to talk about this story, traveling from the gut to the brain and exploring the role of gut-derived metabolites such as short-chain fatty acids in bacterial translocation and in stroke. So we studied stroke. I also did my PhD in stroke, but since I started my postdoc, I am looking from a different perspective. So ischemic stroke derives from the occlusion of a brain artery, leading to brain damage and functional disability. And it's important to know that the stroke affects 19 million people each year, which means that each 3 seconds a stroke occurs. With quickly noticeable symptoms such as loss of balance, face dropping, or arm weakness that I think that all of us know. But maybe it's less widely known that a stroke also provoke post-stroke infections, mainly pneumonia or urinary tract infections that affect between 30 and 40% of all patients and account for 20% of mortality. So it's one of the most lethal complications. And it was traditionally attributed to nosocomial infections or surgical procedures. But however, a groundbreaking study published in Nature Medicine some years ago revealed that post-stroke infections originate from within the host. That means that post-stroke infections will mainly derive from the translocation and dissemination of commensal gut bacteria. So of course, ischemic stroke is a rupture of a vessel and it provokes neuronal death, but it exerts also a systemic response that affects many organs, especially the gut, and it provokes a disruption of the gut barrier and most of the bacteria that are inside. Of course, some to the immune suppression that is also happening can travel to different organs, producing these infections. But however, the mechanisms underlying these infections remain completely unknown, and also the bacteria that translocate or the routes that they follow, they are elusive. And of course, I think we all know that the brain, the immune system, and the gut are really well connected through different pathways, such as the vagus nervous system, the circulatory system, or the enteric nervous system, and of course through different metabolites or molecules such as neuropeptides, inflammatory factors, or gut metabolites. And within gut metabolites, we are particularly interested in the lab in short-chain fatty acids that derive from the bacterial fermentation of some fibers that we can take in the diet. And they are the most common are acetate, butyrate, and propionate. And they are known to have very important health effects. They are essential for brain homeostasis, for microbial development, for neurotransmitter production. But they are also known to have neuroprotective and anti-inflammatory properties in many neurodegenerative diseases, such as Alzheimer's, Parkinson, and also in cerebrovascular diseases such as stroke. It is also important to note that they exert antimicrobial activity in colitis and with infections, for example. And also, it's very well reported that low levels of short-chain fatty acids in the fecal content or in the blood of these disorders, especially, for example, in Alzheimer's or in stroke, it's correlated with worse prognosis of these diseases. However, their role in post-stroke infections remain completely unknown. So with this context, in this project of the Ramon Areces, we wanted to know if short-chain fatty acids are or can be key modulators in bacterial translocation and in the dysregulation of this brain immune gut axis that is underlying post-stroke infections. So first, we wanted to know if they have some potential therapeutic effects in not only bacterial translocation, but also brain injury, gut dysfunction, and immune response. So for this, we performed the MCO model, that is middle cerebral artery occlusion model in mice. So in this model that it's a bit complex, but briefly, we introduce a filament towards the common carotid artery that is here in the neck. And then we occluded the middle cerebral artery for 25 minutes. And after this time, we withdraw the filament, and then we can have the ischemia. And 4 days after the stroke, we take the spleen, the liver, the lungs, the heart, the brainstem, and the blood, and we culture them in brain high infusion plates. And after an overnight incubation, we measure the colony-forming units. It's important that we compare these MCO mice with sham mice that follow exactly the same procedure, but without occluding the middle cerebral artery. So we didn't have any ischemia, but the surgical procedure was the same to rule out the possibility that if we had some bacteria was not due to the surgical procedure. So this is what we got. And here in the white dots are the stroke mice. So what we saw is that we have significant bacterial presence in the spleen, in the liver, in the lung, in the heart, and especially in the brainstem after stroke, compared with sham mice. And interestingly, we didn't have any significant difference in the blood. Also worth noting is that we had also some mice that didn't have any infections, as humans that some of them also don't have. So then once we confirmed it, we wanted to study, as I said, the therapeutic potential of these short-chain fatty acids. So we injected a mixture of acetate, propionate, and butyrate in a blinded and randomized way in 12-week-old male mice after stroke, 24 hours, 48 hours, and 72 hours. And then we sacrificed the mice at 4 days. So this is what we got. So after the treatment with short-chain fatty acids that you can see in the red dots, so we saw that there was a significant decrease, for example, in the liver, as you can see here in the plates. This is after the treatment, also in the lung, in the spleen, or in the brainstem. So, for example, if we focus here, so you see that after stroke there is a huge presence of bacteria that, however, is greatly reduced after the treatment with the short-chain fatty acids. So then we wanted to know if this bacterial translocation was modulated. Of course, this might have some effect in the colon or in the gut as well. So then for this we measure colon length, and then as you can see here in the picture, after stroke the colon look completely different from sham, naive, or even treated mice. So we saw, and this is also reported by others, that after stroke there is a reduction of colon length because there is a gut dysbiosis, but after the treatment with short-chain fatty acid we could partially recover. As you might have noticed from the picture, the fecal output was completely different between the mice, and we counted the fecal pellets, and then we saw that 3 days and 4 days after the stroke, in the treated mice they had much more fecal pellets compared with saline mice, so that means that also gut motility might have been improved. And to study this structure a bit further, we implemented in the lab the so-called Swiss roll technique, but it's not because we are in Switzerland, it's just the name. Sorry for that. So in this technique, basically you take the colon, you roll it, and then you cut it, so you can see at a glance the whole structure of the colon. And then you can study, for example, trayectorias that are these proteins that maintain epithelial cells together and are responsible for gut integrity. So we studied in Thonola occludens 1, and then this is the distribution in green of the whole colon in sham mice. So as you can see, after the stroke, there's a huge reduction of this protein, trayectorian protein, and then after a stroke, after the treatment, sorry, is partially recovered. And so, of course, as we are studying stroke, we are interested in the lesion, in neuronal death, in the brain. So we quantified as well infarct volume in these mice. So for this, we measure with cresol violet, and this is the brain of the mice. So basically sustaining that if neurons are healthy, they're intact, they are staining purple, but if they're dead, they are stained like in fade purple or even in white. They're completely dead, as you can see here in red. So this is how it looks a stroke in brain mouse. And however, after the treatment with short-chain fatty acid, we could see that it was reduced, especially in the hippocampus and in the cortex. It was also very important for us that the survival of these mice was much better at day 4 compared with saline. And of course here we wonder, because we had some bacteria in the brainstem, we wonder if these bacteria also reach the lesion, because this was not reported before. So we perform a staining with peptidoglycan, that is a component of the cell wall of the bacteria. So this is how we look the ischemic core. That means the part where all the neurons are dead. So in yellow, you can see all of these bacteria that they are surrounded by the glial scar, that are astrocytes that are just forming like a scar to contain damage. And as you can see, after the treatment, there was a huge reduction of these bacteria. But something that was very interesting for us is that these bacteria, even in the saline mice, they were just in the ischemic core, that means in the lesion, but they were not in the pre-lesion, or they were not in the contralateral healthy tissue. And if we look a bit deep into these bacteria, so they look like this. This is a zoom in. And if we colocalize them with microglia, with EVA1 or macrophage-like cells, you could see that most of these bacteria were being phagocytosed by microglia, but then some of them were in the parenchyma. So this is 4 days after stroke, and this is very concerning, because if you have some bacteria in the brain, that of course might not be there, and especially if microglia or macrophages are phagocytosing them, that means that the recovery of the lesion can be quite difficult. So what we are doing now, and this is a big work in progress, is that we have taken these brains, and then we are analyzing these bacteria to really know which bacteria have translocated there, and what are the effects. Finally, we also wanted to know if we have some effect in the immune response, because they are known to be immunomodulatory, anti-inflammatory. They have anti-inflammatory properties. So as I said, in a stroke, you have an immune depression after just like a few hours after a stroke, and it can last for days or even weeks. And you might think that maybe that's a bit of paradox, because of course after a stroke you have inflammation in the brain, and that's true, but then like the body tries like to counteract this inflammation, and then it provokes this immune suppression that is also of course helping all these bacteria to spread. And so we measure spleen weight. That is quite good to see this immune depression. And of course, after stroke, we had this huge decrease of the spleen weight, but however, it was partially recovered after short-chain fatty acids. So then we thought, okay, maybe we are going to look a bit farther on this. So we took mesenteric lymph nodes, because of course they are the main place where all the T cells are being primed. So then we decided to look at them, and then we saw that after the stroke, the number of CD45 cells were decreased. But when we look a bit further in these cells, we saw that the number of CD8 T cells or CD4 T cells was almost 0. You can see them here in the graphs. But however, the treatment with short-chain fatty acid, they had a huge response, and then it was almost recovered. So we think that also this recovery of immune depression is helping in the stroke. Of course, we cannot say that it's just like targeting the bacteria or targeting the immune response or targeting the brain. So we think it's like really like a compendium of many things. So for this, the take-home message that I would like to give is like bacterial translocation occurs to many organs after ischemic stroke. This was a bit difficult to believe when it was first published, but I think that is well established now. And also that short-chain fatty acids can modulate fat integrity, brain injury, and also bacterial translocation. So not only in the brain, but also like more systemic effects. So they can be key modulators of this axis. And as future perspective for the work that we are, this is still in progress, but we are very happy about it, is that we are also taking stool samples from stroke patients from the Lausanne University Hospital, And we are with a European consortium with Dr. Adam Sorvia and Corinne Benakis from the Institute of Stroke and Dementia in Munich. We are doing some shotgun genomics of these samples, and then we want to know if we compare stroke patients with infections, if we have some difference in the microbiota. And because it has been shown that in other diseases with bacterial translocation, mainly gut disorders, if you have less of these bacteria in the stool samples, that means that these bacteria might have translocated to somewhere else. So this is what we wanted to know. And it's very, very preliminary, but we look at the relative abundance of bacilli, for example, and then we saw that the patients with infections, they have a reduced number of bacilli. And actually, looking into the bibliography, we saw that bacilli infection is documented, at least in this study, in 34% of all patients in the stroke group, and in the ones that died, it was more than half of them. And also, for example, if we look at the relative abundance of the proteobacteria, we had this very similar trend. And also, we saw that the bacteria that in some stroke patients in the thrombi, they are enriching in proteobacteria, so that might say the same, that if you have less bacteria, less of these species or genera in the stool, it might conclude that they might have translocated to somewhere else. So we are still onto this. And yes, so I would like to add this also take-home message that maybe alterations in gut microbiota profile of stroke patients can also serve as biomarkers for predicting these post-stroke infections. And with this, I just want to thank my hosting groups, Professor Lawrence Hare and Caroline Po group in the Lausanne University Hospital and the university, and of course the founders, especially the Ramon Aretes Foundation and the University of Lausanne. Of course, very happy to collaborate, to take questions, because we're also interested in other neurological and cerebrovascular disorders. And of course, as we are like more stroke-based researchers, we're always looking for microbiologists to help us a bit. So thanks a lot.